All right, so we're going to start here. It's time to talk about sample lineage. You may have heard us talk about lineage here and there in some of the various other talks, and so I'm going to do a little bit of a dive into this topic a little more. This is a little bit developer focused, so if you've got questions about some of the APIs that I'm going to show you, you can come find me later. All right, so what is lineage? So the way we think about lineage in LabKey Server is, typically about deriving samples, pooling samples, aliquoting them. So you may be taking a plate full of samples and putting them onto a plate, and you want to give each sample on your plate a unique ID. So we would use sample lineage for that. There's the hierarchy of samples that can be created through the lineage. In this example, I've got at the very top an expression system. And the expression system, this is something that we have in biologics. And so this lineage is showing samples that are derived from an expression system that may have come from an experiment where you're pulling samples out. And so this is all backed by something you might be familiar with in LabKey Server, which is the experiment run. The experiment runs aren't shown on this particular graph, but they're there in the background. And so this graph is something that we're making available in biologics and Sample Manager, and pieces of it will make its way into LabKey Server as well. So like I said, this is truly a graph, it's not a tree, right? So you can split and pool. And on our little lineage graph here, to make it our first version of the lineage graph, we kind of just put everything on the page. And it turns out looking at a plate of samples doesn't work so well when you're trying to render a graph like that. So we combine them together, the nodes, and at the very bottom there, you can see there's 7 samples kind of combined together as a single object there. So I mentioned that this is backed by the experiment run framework. Another type of run that you might be familiar with, besides just sample derivation, is the assay runs, right? So every time you import an assay into LabKey Server, we create a run for that, and we attach inputs. In this example, we've got some specimen samples. They're inputs into the run, and the little orange hexagon in the middle represents the run itself. It gets the name of the assay file that you uploaded. And then down at the bottom, there are 2 output files. One, so the way to think about it is the samples go into the machine and a file comes out, right? And so the file is the output of this process, of the assay process. There's another node in there. There's another output, which is the transformed data file as well. So when we look at it at this level, the samples come in and some files come out. But we can go deeper. And we can look at the internal structure of this run in a little more detail. So the samples come in. There's a first step, which is to essentially capture the fact that we've imported the data and we produced a file. Then a second step is run, which is, in this case, a transformation script called transform.jar. And that produced a second output file. And that file is used to import the assay run results into LabKey Server. But we mark both of those files as outputs of this process, just for consistency with other runs that do not have transform scripts. So lineage is the experiment runs. There are lots of different types of runs. I just showed you kind of sample derivation or assay, but there's many, many other types of runs. There's MS2 runs. There are file-based script pipelines that can generate runs. There's some other examples as well. So essentially, a run consists of a protocol, some protocol that describes what your run is about, right? That kind of describes the structure, maybe. So that there is a flow protocol or an MS2 protocol. There are inputs, and those can be either data or samples. And data, in this case, means typically files, right? When you're doing an assay import, those are typically files, but not always. So for example, in that first lineage graph I was showing you, there was an expression system that was an input, and that expression system is backed by an object called a data class. And so that data class is a row kind of similar to a sample. It has a name, it has some properties. It's not a file though, and it is used as an input into the run. There's a series of steps. Typically, like for sample derivation, there's one step. For assays, there's one step, but there are some more complicated processes that you can capture within LabKey that have multiple steps, right? The MS2 runs, you could do fractionation, you can pooling, and you can describe all of this within LabKey Server. And we don't expose a lot of that in the user interface, but just so you know that it is possible to capture that kind of information. All right. One other type of run, yeah, I should mention is the ETLs can create experiment runs as well. And so that's another way of capturing a bunch of different steps that may have intermediate transforms. Okay. There's lots of documentation on our labkey.org website about the terminology. This is a very pared down list of the terminology that we use for the experiment framework: protocols, inputs, outputs, and steps. There's protocol applications that capture the time point at which that protocol was applied, things like that. All right. So how do we create the samples lineage? I'm going to focus on sample lineage, but there are other types of runs I was talking about. So you can go to LabKey Server UI. If you go to the detailed page for a sample, there's a little button that says Create Derived Samples. Or on a samples grid within LabKey Server, you can select a few samples and hit a Derive Sample button. Or you can use the brand new Sample Manager UI and Biologics UI to create derived samples through that interface. They're backed essentially by the same API within the server. And there's some other ways to create lineage. You can do bulk importing into a sample set for samples and provide some magic input columns that tell us how to associate a newly created sample with some parents. And a kind of an equivalent version of that is the LabKey Query Insert Rows API for that sample set. And then a little bit different abstraction. You can use the LabKey Experiment Save Batches. These are JavaScript APIs: LabKey Experiment Save Batches, Save Runs, and the LabKey Query Insert Rows. Those the Save Batches, Save Runs began life as a way of importing assay data, but you can use it for sample derivation now as well. And you can indicate which samples are going to be created. They could be prior existing samples that you want to kind of associate with each other, or you can use it to create new samples. And 19.3, save runs. So you can ignore the batch object if you don't really care about that. All right. This is the Sample Manager UI to create samples. So we're looking at a grid of samples. We've selected a couple samples. We're going to click the Create menu item, and you can see there's a list of sample types: sample type 1, sample type 2. These are the target sample sets to create new samples from the samples that we've selected. And so we can see this is our target sample type, but we've also indicated that we want to create a parent column of sample type 1. And so then that corresponds to this sample type 1 parents. And in the value here is a commissary list of all those parents. And so you can do this directly through the UI. You can do that kind of through a bulk import, as Hannah was showing yesterday. The little grid here is kind of cool. It allows you to type in manually or use a barcode scanner or something like that. And it'll do a lookup to find those samples that match the text that you type in. And then, yay, we've created 4 samples. And so these are those samples. We kind of show you the names. They don't have any data here. It's not very super interesting. But what we get out is this lineage chart, right? So we've got the grandparents, the parents, and then our sample, and along with some data for the selected item. All right, so the same, kind of interface that we're doing, that we just showed you, right? You can kind of achieve the same effect by importing a bulk TSV file with providing these magic material input / samples. So this is the sample set name, samples. And this works for other types of data as well. So we got another material inputs and a sample set called mixture batches, or a data class called data inputs with a data class called cell name, right? So we can kind of mix and match. So we've got a Cho-K1, this will be our parent, and this is our cell sample one is the sample that we're going to be creating linked to that parent. And then likewise, so for down here, we got the cell sample one, sample A. So I just wanted to show an example of, even within the same import, we're going to find this cell sample one, which was created here, right? So we can kind of create a long hierarchy of lineage, all in one import. And this batch identifier should have been in this column, but you get the idea. All right, so again, just kind of another interface for doing the same thing. So this is an insert rows call. And we've got our array of rows here. In our first row, I've got example 1, and in my second row, example 2, I've got a column, material input / samples, the name of the sample set. And then I've got my name of the sample there. So it'll do the same thing as you saw in the previous slide. And then finally, save batches or save runs. It's a little more complicated, so we've got LabKey experiment save runs. We need to name that protocol, like I was saying before. In this case, it's the sample derivation protocol, but you could provide an assay as well, right? So if you wanted to import assay data, you can use the save runs API. I need to name my run. If you don't, it will just generate kind of a date stamp or something like that. And then you need to list the inputs. There's an existing one with ID 105, or we can get it by name, expression system 1.205, from this expression system sample set. So we need to kind of know where this name comes from. The names of samples are unique within a sample set, not across all samples within the system. And then finally, here's our output sample, example 3. And we're going to place it into this sample set called samples. And here are some properties of that sample, right? So you could have more columns here. So these are kind of 2 or 3 different techniques to do the same thing with kind of different ergonomics. All right, so how do we access the lineage? We've created some samples. What do we do now? So LabKey Server has that run graph UI. You may have seen it when you click on a run details for an assay or something like that. But we have the new improved biologic sample manager Lineage UI that we've been showing off. But as a developer, you may want to access that lineage directly. There's a couple of ways to do that. You can use the preferred approach at the moment is to use the LabKey experiment Lineage API. And so this will, I'll show an example. You hand it a sample name or an internal identifier called an LSID, and you'll get back a graph of all the nodes, and that kind of lets you traverse that graph of parent and children relationships. It is possible to access the lineage using query, but it's kind of a limited form. It's not as rich as the Lineage API. And there's some other caveats I'll get to in a moment. And kind of lastly, you can also filter the grid using Lineage, which is kind of fun. All right, so here's the Lineage API itself. You provide this internal identifier LSID. So LSID is typically a hidden column on the samples or data class grids, but you can access it by using a select rows, or you can add it to your grid and you can copy and paste it, whatever. So this internal identifier is unique. I was saying that the name of the sample is not unique across the entire server, but it's unique within a sample set. The LSID is unique across the entire server. Then you can indicate the direction of lineage that you're interested in. I want to get the parents. I don't really care about the children right now. And I want to only go 3 generations back. If you leave off the depth, we'll traverse the entire lineage all the way back. All right. What else we got? We also have some more hidden columns on sample sets and data classes, inputs and outputs. They are kind of these pseudo virtual tables with, so we create a foreign key to this lineage, essentially. It's available on run materials, which are samples, and also data class tables. They're a little bit challenging to use, I have to admit. They're nice because you can see the lineage within the grid, and the grid is kind of our primary user interface. The challenge is they're multi-valued foreign keys, right? So if you want to go get a parent, there's more than one parent. I have 2 parents. And the value that comes back is just a comma-separated list. We do a little bit of tricks, to make the links work so you can click on the name and navigate to see those things. But in general, it doesn't allow some other functionality that might be useful, that might be used to in other foreign keys. You can't traverse additional hops from those multi-value foreign keys. It just kind of breaks the query relational engine. There's also performance implications. We've spent a lot of time kind of optimizing the lineage. So initially when we, I don't know, before we started biologics, when you access the lineage, we would kind of traverse the lineage one layer of runs at a time. So it was very iterative and it was very slow. So we had to go query the database, pull in a bunch of data, inspect it, issue another pile of queries, and then kind of continue, right? That was very slow. We early on decided that we would need to shove that into the database as much as possible. So the way we do queries now for the lineage is we kind of maintain a relationship within the database as much as possible to traverse the parent-child relationships. That works as great. We spent a lot of time tuning that. But then viewing that within a grid, I mean, if you think about it, right? So you've got you're looking at a grid of sample sets and you pull in a lineage column. You're essentially asking the server, give me the entire lineage for everything within the entire set of samples that I'm looking at. And so that, but then when I render it onto the page, I only care about the top 100 rows because that's what I'm looking at within the grid. And so we were spending a lot of effort calculating lineage for the entire sample set, essentially, but then only looking at the top 100. So as another performance tweak, what we did, we're kind of delaying when we access that lineage to render time as much as possible, so as late as possible. And there's some implications. That means that sorting and filtering is not possible. Sorting and filtering happens at the database level. So instead, what we're doing is we're row by row accessing that lineage and kind of rendering it into the HTML as we go. And so this is what the lineage columns look like. Click the Show Hidden Fields, you'll see a new Inputs and Outputs node, and I've kind of expanded it a little bit. You can kind of traverse this little hierarchy of virtual nodes. I'm interested in samples, so I expand the Materials node here. Sample and Materials, kind of synonymous. This is my, I can access the entire, the lineage for all samples, or I could only ask for the first generation. That's what this first means. And then we also have, for every sample set, we'll create an additional node. So I have only one sample set called samples in this example. So once I click that little check mark to add it to my selected fields, it'll show up here on the selected fields panel. And then so we can see the selected fields. The input materials first, these are my parents. Outputs material first are my children. But yes, we don't have to stop at one generation. We could go many generations for each of these samples. All right, and finally, the last little bit I want to talk about with the lineage query filters. So one of the fun things that we can do here is, now that we have a lineage relationship, we can filter the grid by that lineage. So we can say, given this node, I just want to see all the data for these children. And so this is what this filter does. It's a kind of a special filter. So I'm sure you've noticed every time you change a filter within LabKey, we'll stick a filter clause on the URL. So the filter clause looks something like this. Query is the name of the data region. LSID is the name of the column that the filter is attached to. And then this funny tilde, and then the name of the filter, exp::child of, indicates I want to do this lineage filter. And then the parent LSID. So I'm interested in the child of all the children of this parent, right? And so that's the fun thing we can do here. And then we also have an equivalent is, you know, the opposite direction. We have a parent of as well. Demo time. Ooh, that worked. Make sure I'm logged in. Okay, cool. So this is a little example app that I started, I think, a couple user conferences ago to kind of show how to customize a samples grid. And so I've extended that a little bit here. So it's, we're going to put this example up in our documentation somewhere. So right now it's just kind of living right here. So this is a little example app, you know, it has a little button that I've added to the button bar to create new samples, and I can say, I want to create a sample within this project and of this type, with amount, and then I can also indicate how many I want of those. And that'll just call the LabKey Insert Rows API to create those samples. I can also select a few from my grid here and say create new. And so this is the new kind of thing that I added for this demo, is to show you that we can create, I'm just using the LabKey APIs again to create derived samples. So I'll just do that. Create 2, 3, whatever. And we can go. Creating samples, woo. All right. So here are my new samples. I've also selected them. I'm going to update the status and mark them as collected so that I can, I'm going to click on one of these guys. And so now I can also, I'm showing how to access that lineage. So I'm using that Lineage API to ask the server, you know, give me the entire lineage, is giving me the entire graph. And I'm just rendering, it is a, unlike Alan's talk, this is not React, I'm sorry. This is just HTML that I kind of splatted together, and very, I ran into a lot of encoding issues with HTML while doing this, so I don't recommend this approach, but it got the job done for this. All right, so then I can look at the lineage of my sample, and then I can also kind of traverse that. So I'll just make an additional request and traverse that lineage, and you can see this is just a little toy app. But essentially, I'm not really doing a lot of processing on that lineage that comes back. I'm just kind of rendering a little tree view or a little unordered elements of these things. And finally, the last kind of thing I want to show you is the parenting. Let's see here. I can select a sample, and I can use that filter. And so now I've added a little filter, and it says, is child of, and then it shows me the name of the thing that I've selected. And these are all the samples that are children of that. And so I can see the status, and so it filters down using the lineage based on that hierarchy. It doesn't, I think the only caveat of that is showing me the entire, all of the children. It's not looking at the depth necessarily. So we could maybe change it in the future to say, I only am interested in the immediate children or the immediate parents rather than the entire lineage. That's it for my demo. Any questions about that so far? So right now the Lineage API only works with a single node. So I can only do one at a time here within this chart. But that's just because that was the initial use case. Yeah. There's nothing to stop us from looking at two samples. Are you saying you want to look at like the intersection of those two lineages, or are you just return all of them? Mm-hmm.
Like we have all the way to the end, have all the tuning clones, we want to know parents per parents. But we have 8 of the tuning clones.
Right. Right. So the question is, can we access the lineage for more than one sample at a time? And it's possible, just not with the current API. Yeah.
So if you, in your previous grid view, select the first children or parents, right? Keeping off all the way because
Um,
grid, you can add a column that shows the parent or children
yes.
first.
Yes.
When you select all, it will separate different, like the comma separate.
Yeah, I can quickly show that. So the question is, can we see more than the first level of sample lineage within the grid?
Right, how does it show it?
And then how does it represent that? Yeah, so let's just try that. So I'm just going to my sample set, and I've created a custom view called lineage. And so this is the first generation, but let's go and customize it so we can see all the generations at the same time. So first I'll click the show hidden fields. I gotta find this column. So let's look at finding all the parents. And so I will, let's see, we're only interested in materials. We'll put the parents right there. So it represents it the same as if it was first, right? So it's just a comma separated list. It doesn't indicate the depth of the lineage within this view. That's why I said that using the Lineage API, it gives you more details about what's going on. Whereas this, you can see here, this is my immediate parent, but then here are, maybe it's my grandparent, maybe it's my, I don't know how many generations back, right? Yeah.
So how does this, the sample manager interact with samples that are uploaded? Like not generated in LabKey, but uploaded from the external sample manager and being kept in the specimen archive or specific study?
Oh, specimen archives. Okay, so, right, specimens are completely separate from sample sets. Yes, not to be confused with one another. I don't know, do we want to say anything more about specimens? Specimens are very much tied to studies right now. They've got a lot of constraints about how they are, you need a specimen ID, a time point and a participant, and vials and events come in, right? Sample sets are a little more open. You can kind of add fields and you can kind of configure them how you like. But there's no, for example, there's no vial parent specimen relationship within sample sets inherently, right? So there's just a sample row. So there's slightly different things. And I think one of our long-term goals is to kind of reconcile the differences between the two. Yeah.
And it makes a huge tree. You display it all. I know you said you collapsed at the bottom of that one, but can you collapse nodes in the middle?
Yes, yes, yeah. They will be collapsed in the middle. I don't have an example of that handy, but yeah, the intermediate. So what we'll do is we'll collapse at each generation. So siblings will be collapsed. Is what I'm saying. So siblings will be collapsed, and then the next generation of 100 samples will be collapsed. So you'll see if you've got 100 siblings from your parent, and then some of those children each have their own 100. They'll each have their own 100 below that are collapsed together. Does that make sense? And
User viewing that uncollapsed and collapsed node, or is that all fixed?
the collapsing right now is fixed, but you can select the collapsed nodes. Let me. Should I try this? Okay. Oh no, I'm running on the cloud. So we'll see what we have on the biologics example here. Let's go look at samples. I only have 25 samples. Okay, let's create some. Target sample set. This is using the old user interface, as you can tell, for sample generation. I'll make 100. Will. Sample type. Blood. Sure. Okay, so I can then go find this guy, look at the lineage. When I look at the child, I just see the hierarchy as just like this. But I can click around, click on this one, and it'll show me there are 100 other, there are 99 other samples not shown in the grid. And I could say, I want to see all of these here, or I could just kind of say, I want to double click this and see if there's anything else going on. And we'll see that collapsed 100. And then we can say, I want to see, let's go take a look at this. So it basically just takes you to a grid view filtered by those samples within that cluster. And then, okay, so for fun, let's take this one and let's create some more samples. Oops, 94. So if we look at the lineage again, let's go back up. So we'll see there's 100 here and then 94 here. Does that answer your? Yes, no? Okay. Okay. Yeah. No, not here. And we have plans to kind of incorporate, so we're hiding all the runs right now. So if a sample was used in an assay and there were some output samples, for example, that would show up here. But we don't link in, say, like if a sample has some assay data, we don't like display that within this viewer. That would be wonderful. Yeah, I think that'd be great. Yeah. We've heard that suggestion before, so it's, yeah. Mm-hmm.
Building on that same question, like sometimes when these things go into assays, they're like technical replicates, biological replicates. That information saved, or would that have to be on that assay level?
Okay, so the question is about replicate samples and grouping them together by replicate somehow? Yeah. Right now, the lineage graph only groups by level, but I could see some other kind of grouping options as well. Yeah.
I would add to that that technical replicates are probably going to be represented in the assay or biological
Cool. All right, any other questions? Yeah? It is. You can make a cycle, yeah. I don't recommend that. The Lineage APIs protect against that. So the question is about if this is a directed acyclical graph, can I make a cycle? Yes, you can. Typically in practice that doesn't happen, but the Lineage viewer and the Lineage APIs will kind of detect the cycle and stop recursing. All right, cool. Oops, that's not what I want. I want to go back. Right. Huh. Okay. Oh, extra content. We went too far. All right. So on our labkey.org website, we've got lots of documentation about experiments, lots of documentation about deriving samples. And we'll hand out these slides so you can click on these links. But You can also search for these topics, and the search works. The sample status demo that I showed you, right now it's kind of the old version at the moment. I'll be updating that within a week or so with the new enhanced version so you can see. Yes, a question?
On the, it's probably answered in the API, but the save batches, I had a question of when will it overwrite, and like when can you force it to overwrite, and when is it, or is it gonna just populate everything?
Okay, yeah, so the question is about using the save batch API, will it overwrite the lineage? Or, okay, yeah.
If you're processing in different batches, when will it overwrite, and when will it keep it as, or just keeps everything as new?
I need to double check, but I think, so for the most part, we'll keep the lineage as is unless you tell us that it is, I shouldn't say that. I don't know. I need to follow up on that. Yeah, whether the lineage will be completely recreated if you have the input and output samples. I know that during the bulk import of the TSV, what we do is we try to preserve that lineage as much as possible. So for example, if you're updating a row and you're pruning a parent or something, you say, I screwed up. That's only derived from one sample, not two samples. We'll remove that sample from that lineage and create a new run. So kind of, because that other sample may have been used elsewhere, right? So, and so here are some JavaScript API docs. There are Java versions of these APIs or Python or whatever, but usually our JavaScript API docs are most up to date. So if you have any more questions about using those APIs or anything like that, come find me, and yeah, we can walk through it. And just for fun, I wanted to show. This is from we have a Czar tutorial. Czar is a fancy word for experiment archive. It's a giant pile of XML. When you import this giant pile of XML into your LabKey server, you'll get a pretty picture. And so this is showing you a case and a control sample going into a run, a giant run. And then out comes 5 samples, MZXML files. If you drill into that run, you can see that the run has a lot more structure. This is all described in that Czar XML, and it's quite a mouthful, but it's showing us intermediate steps. It's showing intermediate samples that are not produced as the final output of the run, but are used as a part of the experiment, as well as intermediate output files. Like a chromatogram file. And then the final outputs are down at the bottom marked in bold.
So
But
are you saying if you exported a bunch of sample and assay stuff into a Czar and re-imported it somewhere, we actually retain all of them?
yeah, you can export and import Czars. So it's a little bit low level sometimes, but yeah. So if you go to an assay run within LabKey Server, and you expand a little export tab on the panel, the panel will show, the normal export to TSV, export to Excel, and then there's also a new tab that says export to Czar. And so the Czar XML is kind of used as a way to export that and import it into another folder or into another server. And when you do that import, it'll create samples for you in order to preserve that lineage. All right, what else we got? Here's another one. This is fun. This is 2 runs, got to kind of stitch together in a graphical program, but you can see there's 2 little A's. And so this is a RNA-seq matrix. So this was generated using a script pipeline, which is another feature in LabKey Server. So the script pipeline takes an input file, runs some, in this case, an R process, and spits out some files. Then those output files are imported into LabKey Server. And so this is kind of describing that whole process, at least the assay import side. And yeah, that's about it. And that's not interesting. All right, cool.